Hi, my name is Brooke Tawit Maru, a PhD student at McGill University, and I would like to take the next 2 minutes to give you a bit of context about the research I'm presenting here and why it's important. Acute myeloid leukemia is an aggressive cancer which affects more than 1,000 Canadians yearly. The current induction therapy is Cytarabine, which is a nucleoside analog. But approximately 80% of patients eventually relapse, and sadly, we don't have an effective salvage regimen. Therefore, our team has been working to understand the mechanisms responsible for cytarabine selective killing of white blood cells in the hopes of providing more targeted personalized therapy. The work so far has produced several publications. In particular, the long-standing model of cytarabine's mechanism of action is that it gets incorporated into DNA and inhibits DNA strand elongation, hence leading to cell cycle block in the S phase. But work from our lab has revealed that it's actually the incorporation of cytarabine into RNA primers of DNA synthesis that triggers programmed cell death. In fact, incorporation into DNA rather than RNA primers is actually associated with resistance in approximately 40% of AML patients. So this suggested to us that maybe it's the defective expression of genes involved in this DNA synthesis system that leads to loss of sensitivity to cytarabine. So this project is a continuation of this story where we have now identified several candidate expressional biomarkers, which we're trying to develop therapeutic oligonucleotides to modulate their expression and reverse the loss of cytarabine sensitivity. If successful, we envision a personalized medicine approach where patients with low sensitivity to cytarabine are identified at time of diagnosis and a suitable RNA-loaded lipid nanoparticles prescribed as an adjuvant therapy that increases the efficacy of cytarabine. Thank you.